There are many diseases in the world that are currently incurable, such as Parkinson's, AIDS, and Alzheimer's. The difference with Alzheimer's is that the other diseases do not adversely affect the brain, but with Alzheimer's, individuals start to lose their memories and even the capability to complete basic tasks. That is the reason why it is the focus of our research. There have already been studies done that have isolated the APOE4 gene, which is found only in humans. These studies found that this gene could be a primary factor for Alzheimer's. As a result, the focus of our experiment is to insert this gene into other organisms in order to see if it will cause this disease. Scientists have established that the APOE4 gene is in mice, but mice aren't good human models, so therefore we will be using chimpanzees, which are more genetically similar. There have already been previous experiments using chimps for Alzheimer's research. The way we will insert this gene will be using CRISPR-Cas9, a genome editing tool that involves enzymes. We can use CRISPR because scientists from KAIST have already used this tool to edit the genomes of chimps. We will have two groups, a control group and an experimental group. We will use around 10 sets of identical twins for this experiment because they will be genetically identical and any manifestation of Alzheimer's will most likely be a result of the inserted gene. One twin will be in each group. Both groups will be in the same environment and in captivity, so external factors will not be included. The control group will be chimpanzees that are not genetically altered by CRISPR, whereas the experimental group will be chimps that have had multiple copies of the gene inserted. Multiple copies will be inserted to increase the chances of mutations occurring. This will be a long-term experiment and there will be 3 behavioral checkpoints: a young adult, middle-aged, and elderly. We will design memory tests to see if symptoms appear and then directly compare those results between each set of twins. We will also monitor their behavior as they age to see any changes in normal schedules. To conclude, there are 3 different outcomes for this experiment. If there is not an increase in the prevalence of Alzheimer's in the experimental group compared to the control group, then the APOE4 gene is not the only factor that causes the disease. If the experimental group does show considerably higher rate of Alzheimer's in comparison to the control group, then the inclusion of this particular gene increases the chance of manifesting Alzheimer's. If every single chimp in the experimental group develops this disease, whereas the control group remains at a low rate, then we can infer that the APOE4 gene is the only cause of this disease. With the knowledge that can be gained from this research, scientists can understand more about this particular gene and how it can be possible to suppress the gene if it is the primary factor for Alzheimer's.